Good morning. Such a beautiful morning today, isn't it? Bright and early? No? Research is in the air? Okay, too cheesy. So, Pigeon always jokes that, research is in triple IT's water. If you drink water here, you're already a researcher. That is, I think, truer today than any other day in the year. So this, I welcome you all to the R&D Showcase 2022. We have a lot of, as you guys all know, posters from research students lined up. A lot of hard work has gone into it. You guys know that better than I do. A lot of demos are also there, which are very interesting as well. So I ask you to all, after the inaugural session, please browse through those posters. Now, for the inaugural session, we have Dr. Vinay Nandikuri, who is the director of CCMB, which is a CSIR lab. And it's a great honor to have him. He's a perfect person for this R&D showcase. I'll tell you why. Because the theme for the R&D showcase this year is Tech Away, accelerating into the future. Okay. Now, what do we mean by that? We mean by tech away is that there is technology driving us, accelerating us into the future, particularly after seminal life-changing events like the pandemic. Okay, so things like the pandemic are also sometimes, as grim as they are, can actually be drivers of technology as well. Okay, so that is what we mean by this theme, and that is why Dr. Nandikuri is we have a privilege of having him here for this particular theme. Before his talk, I would like Professor Pijen to come to the podium and quickly welcome us all as well and share his thoughts on the occasion.
Okay, good morning to all of you. Good morning to another edition of this R&D Showcase. I remember somewhat painfully now that 2 years ago, our R&D Showcase in all its glory, which is probably 2 to 3 times the size of it getting organized today, with every student, this whole building and few other halls in the campus being fully taken over for the demos for Saturday and Sunday. That's the R&D Showcase format from 2002 onwards. We were all set to do that on, I think, March 7th or 8th, whatever was the Saturday, Sunday in 2020. And that was March 2nd, Monday, March 2nd was the day first COVID case was reported in Hyderabad. And on third morning, newspapers were full of this and that. And already, of course, there was fear in the air of across the country, across the world. So that was the first event we cancelled. I mean, that's the first event affected by this COVID-19 pandemic in 2020. We said it's not safe to hold it. You know, typically 2,000 or more students and professionals come from across the country, across the city, sometimes from outside the city, also many from outside city come. We don't want to be known to be, a super spreader event, all those terms we hadn't even heard of until then. Super spreader event. So we, in fact, of course, today it all looks so ridiculous after 2 years. We thought this thing may be over by, soon enough. So we postponed the R&D Showcase by a month. I mean, it looks we are all laughing now when we think about it. Anyway, I think the story is academics continued in reasonable form. Research continued fairly well. So I think we should all celebrate the resilience. We cannot prevent these kind of things happening. Our world has too many things in too many corners, and it's too connected today. I mean, if there is a new variant reported in, say, South America or South Africa, surely enough, it will come here because we are very well connected. It's a good thing about the world. So back to R&D Showcase or back to IIIT Hyderabad, we call ourselves a research university. And even the undergraduate students here, undergraduate curriculum is very research-led, very research-driven. Every course, we would like the student to experience something that is not just a routine assignment or known thing, something that is beyond the questions at the back of the textbook. And that's what we mean by research-led. And more than half of our student body are research students, including the undergraduate, the dual degree program where the undergraduates get a BTech degree and a research-based master's degree for which they publish, they defend the thesis, write a thesis, defend it, and so on. So, like Aftab said, research is in the air here or water here. So, and the purpose is very simple. For students, it's not because we like research, but future students or future leaders in technology or science need to go beyond what is in the textbook. They need to know how to solve unstructured problem, how to give faced with a problem, how do you find the solution, or how do you go about finding the roots to the solution. That's what is research is. It's not that you solve all the problems. Okay, so this has been the motto of the institute from the beginning. We had these dual degree programs almost from the first batch itself, and very strong masters and PhD programs, which also have thesis, strong thesis and publications and system building. We have our own incubator technology of the institute being taken to the society through startups or other mean technology transfer. Incubator starts in 2008, one of the oldest incubators apparently, and we are also told one of the largest academic incubator in the country. We have 125 or so startups. And we also decided, since being a small institute, our incubator, if it focused only on our students, alumni, and faculty, it will remain small. So we opened it to the entire city, and we have programs running for startups from anywhere. So this whole ecosystem of creating technologies and solutions in the labs, but also trying to take it forward to the society. This has been our dream, our aspiration from day one. I think as we become more and more known, as we have better and better, research body here, like the students assembled here, we are achieving more. So that's the story of the institute, and we bring it out through R&D Showcase annually. One of my colleagues used to say this is like the Sankranti of IIIT, where our harvest festival. The harvest is our research, and the research output is definitely is important. But I think equally important is the enthusiasm that the student researchers show, because it's ultimately we are an institution which produces lots of students, and they should carry the research message or the fire or the small flame of research inside them, the enthusiasm. It's not only about what they produce, which may be small, but that the small flame that they carry will result in much bigger dishes being cooked through their lifetime. So that is why we are a research-led university or research-based curriculum we follow, etc. So 2022, we are hybrid. We are not really fully back to normal. Since we have 2/3 or more of our students on campus, it is open to everybody, open for physical visit to everybody on campus, and it is available online. Like last year, it was fully online. That is available on the portal. This year, we also have a metaverse app that somebody can install called Next Meet, and it's one of our startups or one of the startups associated with our CIE. Next Meet, anybody who wants to download it from the appropriate Google or their own site, you could go and, I saw a demo yesterday. You can go to a stall, just like you can walk to the stall. You can go in there and ask questions, audible questions, like audio questions, and they'll reply. The person standing there will reply. So it's kind of a small simulation of what it is to be here, but it is available for browsing over a browser from now till, last year's one is available even today. So this year's one will be available too for a long time. So that's the advantage of being online. Things don't go away after the event ends. So I'm very excited to be again, be at the inaugural function of yet another one. And I'm sure all of you are also, and I'm also as eager to listen to Dr. Vinay about his thoughts. I mean, I think the biology or healthcare related research in India got a huge recognition. I think they were all doing great. We found we have so many vaccine manufacturing companies, etc. I think they didn't get the respect they deserved. I think pandemic definitely brought them to the fore, and CCMB and all the associated institutions have been at forefront in Hyderabad, as well as the national force that fought with this COVID in the last 2 years. So we are very pleased to have him here as a keynote speaker, as a plenary speaker for this event. I'm looking forward to that also. I welcome all of you to this event. Enjoy the physical event late for the rest of the day and the virtual one. And write to the students or others involved if you want to know more. And if you want to be part of those things, write to them and say, how can I do this? How can I be part of this? That's the goal also, selling the research to our own internal students. So welcome. Enjoy the event. Thank you.
Thank you, Professor Pijan. Right. So, without further delay, I would like to call Dr. Vinay Nandikuri on stage. He is the current director of CCMB, which is a CSIR facility. Dr. Nandikuri did his master's in biotechnology at IIT Bombay and PhD in molecular cellular biology in IISc Bangalore. He then did his postdoc at Texas A&M and University of Virginia before coming to India in 2004 to establish his research group at NII New Delhi. Dr. Nandikuri is a well-known molecular biologist who has contributed immensely to delineating the signaling networks in Mycositum tuberculosis bacterium, which is basically the causative agent of tuberculosis. He has over 70 research publications to his name in reputed international journals. He holds the prestigious J.C. Bose Fellowship. He is an elected fellow of the Indian Academy of Sciences, the Indian National Academy of Science, and National Academy of Sciences in India. So please, Professor Dr. Nandikuri. Yeah.
Thanks for inviting me for this R&D showcase event. I was just now mentioning to PJN, right? That's how they mention you. I was to call you with full name Narayanan, but that the boundaries between healthcare, the IT, and all these things are kind of going away. If you nowadays, for example, crystal structures, there is an artificial intelligence which actually gives you the structures of proteins, molecules. They make predictions, and the predictions are pretty good. So this is the future. But I want to start with the anecdote because one of the things which he was mentioning is about the water. So research in the water, very good. At some point of time, way back in 1990, when I was a student at IIT, all of a sudden we started getting a certain smell in the water that we were drinking. And later on, we got to know that for some reason from somewhere, phenol got leaked into the water. As long as it is research, not phenol, it's quite fine. So we got tanker water for 2-3 days, and eventually it got fixed. And the phenol contamination was found to be something like 100-fold higher than what it is supposed to be in the water. Anyway, that's interesting. So I like the concept of research in water. So what I want to talk about is the technology is what has changed the way we interact. And it actually, in one sense, helped us not lose 2 years of our life. Without that, most probably, had this pandemic come, say, 30 years ago, most probably a lot of students would have lost a year minimum, if not more. That's for sure. With online classes, may not be optimal. And most probably a lot of teachers don't like it, with me included, because you can't see audience. Without seeing audience, it's never the same. Uh, Without the technology, however, people would have lost a year, maybe two. Um, in that sense, technology, nobody can recognize the value of technology as the students at this point of time. And the connectivity and the way we conduct meetings, the way we do things has changed. And I don't think it'll ever go back completely to the way it was 2 years ago. But I'll tell you, it's still a privilege to really interact directly with the audience. You can see the faces, you know whether they're getting you or they're lost and sleeping. That's the basic important thing. What I'm planning to do today is what I'm going to do is not talk about much of what I do as a research, as a scientist, but I want to give an overview of how, I'll get to this. So overview of, I want to talk about disease biology from the viral perspective, bacterial perspective, and want to talk about utility of the big data in today's context, not simply about virus, bacteria, but even with respect to humans. The way right now, big data is everywhere. In fact, our problem now seems to be that there are not many good people who actually mine the data properly. That needs to change. And that is where the boundary between an engineering, physics, chemistry, and biology is more or less getting wiped out. It has not happened as much in India. It has happened quite a lot in US and European countries. And we need to reach there, because many people who are in the engineering side, who are capable of coming up with writing programs, understanding, and ability to the smartness to mine the data, to make sense of it, which can be used by the biologist to set up experiments. They're part of the teams in Western world. Like labs have teams of people with different components and aspects. People with different expertise. That needs to become part of our institutes also. And I believe that is the future. And it's not very far off. And even I believe this, and from the CCMB context also. Just for the sake of taking you back to what you guys may have done at some point of time. Should I? Something has happened. Okay. I'm looking for a pointer actually. I had to put this. Okay. I'll leave it. I'll use the. So yeah, cell. I want to take you guys back to what our cell looks like. And the reality is both bacteria and viruses use our cell to survive, to replicate, to become something else. For example, our cell has many organelles. And this is the nucleus where our genetic material is kept. And the nucleus then gets converted to what we call as an RNA, which then moves from nucleus to the outer part, which is the cytosol. In the cytosol, it makes proteins. And what we call this in biology is called central dogma. DNA gives to RNA, and RNA gives protein. So this in 1950s was considered sacrosanct. Central dogma. DNA always gives RNA and RNA gives protein. And then came the variations. Viruses are the ones which bring in quite a lot of variations. And viruses have reverse transcriptase, which can make DNA from RNA. So RNA can become DNA again. And that is the first variation of the central dogma. Oh, you have DNA going to RNA, RNA going to protein. But then comes RNA becomes DNA. And then a little later came the other variation where RNA in itself is the genetic material. And like some of the viruses use RNA, they go into your nucleus, become DNA, get integrated into your genome, like HIV, and eventually make more of themselves and come out. They use a lot of our machinery. And then you have other things like coronavirus, which actually make RNA from RNA. So they don't even go back to the DNA. They make RNA is used as the genetic material, and then RNA is converted to more RNA, and they come with their own enzyme. Right? So one of the things about all the viral infections is they use our machinery. They use what we have in our cell to survive and make better. So the thing about viruses are they're non-living as long as they don't have a host, in the sense that basically are not replicating and not doing anything. Once they get into the host, which is our body, they start using our machinery to replicate, spread into other organelles, and it's a fight between them and our immune system. Eventually, depending on comorbidities, depending on many other things, the complications are going to vary. So that's the concept. And if you really think about, if you think that only corona is the one that has come and now we are all having a major viral infection, just let me tell you that there are approximately 200 viruses that can cause cold or upper respiratory tract infections in our body. 200 viruses. And this is not the first SARS-CoV-2 virus to reach our shores. There were COVID, or the SARS-CoV is there in circulation for a while. Quite a lot of it simply causes common cold, right? So what we know is only the superstars, but there are many actors and side actors. There are so many viruses going around which complicate our life. That is not sufficient. We also have bacterial infections, right? So in the context of our body, at any point of time, you are going to face some infection or other, and there is a way to fight something or other from the context of the healthcare sector, which is either you provide a vaccine, or you should have a drug that can eliminate the replication of the virus in your body or the bacteria in your body, an antibiotic in the case of bacteria, or you ultimately have to have some way to contain the things, contain the infections, maybe by boosting our host or some of the other ways to do it. But these are the two aspects, and it is not anymore what it was at some point of time. So just to, again, these are all simply to give overviews. Viruses come in many shapes and sizes. You can see them, that they have double-stranded RNA, double-stranded DNA as their genetic material. They come with single-stranded DNA as the genetic material. They get RNA as the same, positive strand RNA as the genetic material, negative strand RNA as the genetic material, right? So positive strand RNA can immediately code for proteins. Negative requires to be converted. So you have all kinds of genetic material used by viruses, which is what makes it difficult. And many of the times, just to give you guys a feel, our human genome is approximately 10 to the power of 9, 3 or 4 into 10 to the power 9 bases. There's a long length. The HIV virus, which causes AIDS, is approximately 10,000 bases, 10 to the power 4. And coronavirus that we are talking about is 3 into 10 to the power 4. And many of the viruses are in a range, and it is considered a large virus. So they are in the range of 10 to the power 4 to 3 or 4 into 10 to the power 4, maybe at best 10 to the power 5. They're not very, very large. They're very small. Bacteria go up to 10 to the power 6. Like Mycobacterium tuberculosis, which causes tuberculosis, which I'll talk about it in a while, goes up to 4 into 10 to the power 6. So even at a minimum, we have a variation of approximately between 10 to the power 2 to 10 to the power 3 at the level of our genome size and theirs. And these small things can bother us in a big way, right? So what are the major infections that we know of in the last, say, 100 years? I'm not talking about every infection. I'll talk about something that is like superstars of infections in one sense, like HIV, human immunodeficiency virus. When it came, it was big news too, except that the human immunodeficiency virus was not transmitting through aerosol, means through the air, nor was it transmitting through simple touch. Since it was essentially sexually transmitted, or bodily fluid exchange, either through injections or something like that, it was not, it was simply not viewed the way currently coronavirus is viewed. It is as problematic as you have today's viral infection, that is coronavirus. HIV actually enters our body. The difference is it goes into the nucleus, integrates into our DNA. Our DNA, it becomes part of that. And it makes more copies of it and ultimately comes out and then starts infecting. The other problem with HIV is it actually attacks our immune cells. So in our body, we have B cells and we have T cells. T cells are basically, both B cells and T cells are adaptive. And then you have other set of immune cells called macrophages and all. So initial set of response is innate immunity by macrophages and neutrophils and other things. And once you have certain level of memory, presentation happens, or this is a bad one, figure out, B cells and T cells come into play. So these are adaptive immunity. So what happens is HIV actually eliminates the T cells by, it actually gets into the T cells. Because of that, what happens is that over a period of time, your immunity gets compromised. So you don't die because of HIV. You die because of other infections that come because you have HIV. So the problem with viruses is they mutate very fast. HIV specifically has a tendency to mutate too. And because of that, HIV is not treated with single drug. It's treated with a combination of 3 drugs called AZT. Okay, now people don't talk about it. And let me tell you, AZT treatment, while it is effective, it does not eliminate the HIV. It's a lifelong treatment. Once you are infected with HIV, you are stuck with AZT treatment for life. The advantage is viral titer comes so low, you're not infective to others and you survive, but you have to take the drug. You are off the drug for 4 months, you're back up. And the reason why they give 3 drugs is because if you give 1 drug, virus evolves. So a combination is given. That is a human immunodeficiency virus. The second one, which all of us know, but don't worry anymore much about it, is called human influenza virus. Everybody gets a flu. Most of it, not as prevalent in India, but flu is quite prevalent in Western world. Every winter, if people don't take vaccination for the flu, they end up getting it. And flu kills people. Mind you, it's not the flu doesn't kill people. Now it's become part of the life. But at some point of time, way back in 1918, there was a pandemic called Spanish flu. It's caused by this. Spanish flu came in 1918, and the thing lasted for 2 years. And it came in total 3 waves, and it killed 4 crores. That's a lot of people across the world. And this was at a time, most probably this count may be wrong too, but 4 crores, that is the number of people it killed. By the time it was done 3 years later, that is the number. And it came back once again in 1956 in US. And so it became from what you call as a pandemic, which essentially the definitions are interesting. Pandemic is something that's all over the world. Endemic is to smaller areas. So now when people start saying, oh, now corona is going to become endemic, means it's not going away. It's going to be in different areas at different times. That's all that's happening. So unfortunately, once they come, they don't go away. Ultimately, our immune system learns to live with it. And these things continue to be perpetuated. And it is important that we take care of ourselves and we take care of our health. And always precautions are important. Like when you get a certain symptoms, over a period of time, what will happen is you will also have drugs, which are already, one of the Pfizer has developed a drug for coronavirus. So these drugs, in addition to the vaccines, would help in mitigating the viral loads even if you get infected. So you will go into multi-pronged approach to contain this virus. Is this the last virus to see our, is this the last pandemic that we will see? Unlikely. There will be more, and this is going to be how it is. But our hope and the basic thing that I basically want people to appreciate is any kind of a vaccine generation used to be 7 to 10 years work. What you need to appreciate is when the corona came in calling, vaccines came in within a year. Whether it is an inactivated virus vaccine that is produced by Bharat Biotech, where virus has been inactivated, or an mRNA vaccine that is produced by Pfizer and Moderna, or subunit vaccines produced where subunit vaccine is basically you put in only a part of the protein and other things. Subunit vaccines. Also, we have DNA vaccines that were delivered and multiple ways, multiple things. But vaccines have come in 1 year. That's a stupendous and a very major achievement. Had it not been for that, our death rates would have been much higher. And if you want to consider that, like really when Delta wave came calling, the vaccination rate in India, because just we started, was 2%. And you could see the impact of Delta on our population because we were not vaccinated, not previously exposed that much. Even if you're exposed, what happens is virus evolves and it escapes, what we call as immune escape. So the death rate, Delta actually ran through us. None of us were prepared for the scale of that infection. But when Omicron came calling, it was very different. Our population was 50% of our population was by then doubly vaccinated or previously infected, those who survived, right? So that made Omicron wave relatively, it's also added to the fact that Omicron per se as a virus was not as infective as the Delta virus. Now we'll come to that. So when it comes to Spanish flu, coming back to this, how do you contain it? There are two ways people do it. Every year, people are giving vaccines to, you get a vaccine shot. You go to Western world, it's normal. Come winter and they'll give you a vaccine shot. And that is based on the strains that you've seen a year before. Most of it, I don't know exactly how they come up with, but they come up with multi-variant vaccine shot. And also, they have a particular drug called Tamiflu. While Tamiflu is a good drug, it is effective only in the first 48 hours. Have you heard of H1N1? I think few years before, that was in news. The reason it was in news is H1N1 is a variant of influenza virus too, but it was a little bit severe and it was giving you pretty strong symptoms. And Tamiflu was the medicine that was available only through government agencies and all at that point of time. And this would be very effective as a prophylactic. Somebody has an H1N1 in a family, other members can take Tamiflu and escape the infection. So, but this basically essentially stops the replication of the virus. That is why it is effective in the first 2 days and not afterwards. Now, coming to something that we have been facing for the last 2 years, SARS-CoV-2, 30,000 bases, 30 genes. That's all it carries. And it's a single-stranded RNA virus. Genome is only 30,000 bases, encodes for 30 plus viral proteins. First time sequencing was done for this was on Jan 6, 2020. In India alone, we have sequenced more than 2 lakhs since then. India alone, and we were not really at the top 10. Places like England, I don't even remember the number, but huge number of sequencings have been done. A variant comes in South Africa, within 15 to 20 days that the variant is there. Sequencing is biology. Analyzing the sequence is not. That's bioinformatics. Bioinformatics, which many of the biologists have no clue, right? So in the sense, I can't write a program. That is the truth. And so it is the other end, that is you people who have got into the other part of biology that has changed the way these things happen. Next generation sequencing, we are able to do because of the computation power also. the computation powers, the kind of data that gets generated is so huge that we need huge clusters and others to store the data. And you need high-end computers to analyze the data and to ultimately give some meaning to those sequences. That is where we have become a global citizens. We are all together in it now. It's not anymore we do in the lab and you guys are in engineers, and so we are not in the same. We are all in it together now. You guys understand these problems as much as we do. May not be the biology part of it, but certainly the analysis part of it, which we have no clue. But eventually together we make the team. So just to give an idea of how technology and how science actually mitigated the impact of COVID. What did I say that death was with Spanish flu? 4 crores. Means 400 lakhs. Look into the death rate with COVID. It's 60 lakhs. This is world over. Let's assume the numbers are a bit wrong, but that would be wrong for Spanish flu too. So we'll take the numbers as is. The number itself tells you there is something like around 6 to 7-fold difference in the death rate because of COVID compared to Spanish flu, which came 1 century ago. This is technology. This is science. How much science has advanced, that has changed it. Absolutely. Yeah, it's way better. If you consider all the factors, I think we are looking at a difference of around 30 to 40x. Means that much would have been our death rate. Had it not been for our scientific advancement, most probably we would have been at something like 20 crores. Nothing if it were to be the same population in Spanish flu today and no other technology. The other aspect is that, of course, the technology has, the science has changed. The way we look at things has changed. mRNA vaccine technology has come and other things. But whatever it is, its technology has changed, and that is why we are able to survive this. You can't shut down and run anything in 100 years ago, right? We were shutting down, but we were still in touch with each other. Not the most healthy thing, but nevertheless, something is better than nothing, right? So given the kind of situation, grave situation we were in, we could still do things. That is another aspect of technology. So when SARS-CoV-2 came calling 2 years ago, obviously, how do you mitigate a impact of a virus? You need to have multi-pronged approach. Multi-pronged. First thing is you need to be able to detect that you have the infection. That is where diagnosis comes into play. Faster the diagnosis, better is the containment. That is one thing which China has done a tremendous job on. We have done quite good. And to give you guys kind of a feel, the kind of kits when we originally the SARS-CoV-2 came, that for detecting SARS-CoV-2, there were 1 or 2 kits available, and we were importing everything almost. And the cost associated with the test was around 1,500 rupees or something like that. This is literally considered cheap, okay? 1,500 rupees or something was the cost. Today, you can do the test, really speaking, for around 200 or 300 rupees. Okay, and if any of you really want to get a feel for it, if you want an emergency test done in place like US, if you go to an airport in India, you can get it done for 500 to 600 rupees. And they'll give you the results in 6 to 7 hours. In US, the same thing costs approximately $145. So Indians have done amazingly well in terms of evolving and making it a real market force. Now we become the suppliers, whether it is a vaccine or whether any of these kits. Bharat Biotech's vaccine is now also in Australia. And Serum Institute's vaccine is all over the world, which is produced a lot here. So we are actually the vaccine hub. Hyderabad is. Hyderabad companies have produced so many kits which are in market across the world. So what I'm essentially trying to say is that this time, I could see the innovation happening in this in this city. Not as much in Bangalore and this city, a lot more than even Delhi and other places, right? And of course, Pune also. So there are some of our cities and some of the existing kind of force we have, we could do a lot. The second thing that you need to appreciate is this. I mean, Hyderabad, for example, where all of you are, there are 48 academic institutions. I didn't know the number. I was told this by somebody else. That's a huge number. And you have a triple IT, you have an IIT, and then you have AIMS coming up. And then you anyway have multiple research institutes. For example, in Tarnaka, where we are, it starts with IICT, ALS, and then you go NGRA, Geophysics, and then across you have Survey of India, and then you have CDFT. There are 5 institutes in one road alone. Oh, NIN, sorry, 6. So National Institute of Nutrition. There are 6 institutes, and add Usmania, bit away, you have around 7 or 8 in a very small area. That's what Hyderabad has, right? I mean, that is there in Delhi also, and that is there in Hyderabad, I mean, Bangalore too. But this is what Hyderabad currently has, so many institutes. And this area, you have Central University, Hyderabad, and you guys are here, a little bit, I mean, of course, a little bit further away, quite further away, actually, triple IIT. But consider all these things, so many academic institutions, and I'm certainly missing out all the DRDO labs and all the DST labs that are there, right? So there's a huge number of research institutes in this place. But what I'm saying is, one is diagnostics, and we did a pretty good job in sense, not we, me, I'm talking about India. Vaccine, one is produced right here, and there are other vaccine manufacturing. Now we are able to, we had difficulty during the Delta wave, but eventually it was fine. And then you have the treatment options. We have the ability to test and all. So some of them are in clinical trials. Hopefully they will come out. And the major aspects is SARS-CoV-2 surveillance. Like, how do you figure out whether the virus is, what kind of mutants are coming in, what kind of this. So it's huge amount of genome sequencing what happened. Serosurveillance, which is essentially finding out whether you're infected or not by looking at taking your blood and finding out what kind of things happen. Air, what is, whether the air is clean or not. CSR was involved in this. Sewage is because you share the virus, whether you're symptomatic or asymptomatic in your stool. So even sewage surveillance gives us an idea about the virus loads in a population. So in addition to that, we were involved also in COVID mitigation, essentially giving seminars where you are talking about, or you're interacting with the public to make them aware of what we are actually facing, how you can mitigate it. Of course, washing hands, masking up, and all these things. But hopefully, worst is behind us. But anyway, the point is, over a period of last 2 years, we had many major variants that came. And initially, they used to label them as South African variant, Indian variant, UK variant. Later on, they became wiser because it's not a smart move to do, because then the country gets a stigma associated with the variant. The variant is very strong, then it's like, okay, it's originated in India. The virus can originate from anywhere. So we had one variant coming from South America. We had one variant coming from England, one variant coming from South Africa, and one from India, and of course from China. So we have Wuhan, what we call that is also called something else, but alpha, beta, delta, then they get into Greek letters, and now we are at Omicron. So hopefully, while I can be hopeful, it is very difficult to say that. Not difficult. It's more or less given that more variants will come. The question is how, what level, whether it is going to become softer, whether it is going to become more ferocious, and how we take is something depends on multiple aspects, including vaccination, how long the vaccine, how frequently the vaccination has to be taken, once a year. Those things will come as the time progresses. But it is certainly not going to be what it was at Delta wave, because we have a prior exposure. We have seen the enemy, except that they may come back with a beard. So that makes it a little bit difficult. So what is the evolution when it comes to viruses? Virus A gets into your body, comes out as virus B. Why does that? Virus, like anything else, needs to replicate. When you replicate, you make errors, like anything else. Errors are corrected. Some errors remain. These errors, if they are advantageous to me, I'll retain. If they are disadvantageous to me, I will not. Means some viruses die because of these errors. Some viruses which have accumulated some of these errors survive. When they survive, they are going to become a variant. And more they replicate in your body, some of these things can evolve. Suppose you are an immunosuppressed a person gets infected, they can sit and replicate for a longer while because you don't eliminate them as fast. But whichever way, eventually, let us say originally the virus A was recognized by an antibody that is produced because you had a prior infection with virus A, and that antibody would kill the virus A, but it would not kill the evolved virus B because it is not recognizing it because the person has changed the look. The virus has changed the look. So it becomes, so there are again two levels of immunity. Even if your B cells don't work, T cells might work. Whichever way it goes, you do get protection from the severity of the infection, but you may not get protection from the infection per se. That doesn't matter. That realistically does not mean that vaccination should not be done. Vaccination provides that recognition. At least you know the face. Only beard is the difference. At least you'll be able to recognize after 1 hour, 1 day, but you will be able to recognize eventually, and that gives us the protection. This is just to show what Omicron was. This had 35 mutations in one surface protein called spike. It had 35 mutations. That basically meant that many of the things, the existing infection, prior infection, antibodies coming from the prior things were not able to neutralize Omicron, and so it could enter your body. However, because of the other immunity, that's T cell immunity, and even in general, it ultimately was not very effective in replicating. And eventually, the severity of the infection was much low. And I'm sure all of you know that. Does it kill? Yes, it still can kill an uninfected population, those who are never exposed, unvaccinated, and also those who are much older. Remember, the average age the one survives has changed over the last 100 years too, right? And when you are in 80s, your body is not what it was when you are in 60s, and then which is comparable to 40s is bad. So that's how it is. Aging is. So unfortunately, this is the reality too, that we have much older population today than what it was there. And which is good side is that the technology has, the medicine has made it take us there, antibiotics and available treatments. But when something like this comes up, we do become vulnerable. So we in CCMB, this is my only slide on what we have done in CCMB, is that contributed to approximately 12% of all the sequences that have happened in India. So we have sequenced this many samples till now in our facility, and we have established 2 satellite sequencing centers, one in Vijayawada and one in Shillong, so that those 2 states could do their own sequencing in those satellite centers. We were hand-holding them initially. Now they're almost self-sufficient. So that's our contribution to COVID. And in addition to many other things, we were involved in diagnostics, testing a lot of compounds and sewage surveillance and a lot of other surveillance. But last 2 years have been busy. And I'm quite proud of the way my predecessor has taken it and taken this as a challenge and took it to the level it has. I'm glad it is done. And same way, CSIR has also done a lot, including making oxygen, generating oxygen for the hospitals to many areas. Everything was done, including building things fast enough for the hospitals. We were involved in air surveillance and making machines for that, involved in mitigating by putting UV lamps, which will kill many of the viruses and the bacteria in any of these major places. We actually put it even in Parliament, right? So a lot of things have been done. That's viruses. Now, what about the other enemy, bacteria? I work on Mycobacterium tuberculosis, so I'm not going to go more into AMR. At the end of the day, this infection still kills 15 lakh people every year, even today. So if you add the numbers, it will leave all the other infections behind, right? What happens in case of tuberculosis? It is not as much now. At some point of time, you walk into a local train, you walk into a crowded place, somebody sneezes, you have droplets, aerosolic infection, you get tuberculosis. Your innate immunity, the first line of defense, takes it. Like I said, HIV attacks the immune cells, which are T cells. In Mycobacterium tuberculosis can reside in innate cells. macrophages. It stays there. It doesn't get killed by them. It doesn't get presented by them. It becomes something like it can replicate comfortably there. So as our immunity comes into play and they start surrounding the infected cell. This surrounding by neutrophils, lymphocytes, and other things, eventually what we call is a granuloma. It's like something of some kind of a sore, some kind of an pestering wound in your body. But now it is surrounded by the immune cells, hard immune cells. At this point of time, the bacteria has 3 different ways it can spread. One is, of course, your immune cells prevail and completely eliminate the bacteria. The second is that bacteria prevails and you basically get heavily infected. And 10%, around 5% of the cases, people end up getting TB. And the third one is basically where we reach a point where neither infection is prevalent, nor are you, but it has gone into sleep mode, latent infection. It's not gone. Even HIV, when you give AZT, gets into literally a sleep mode. Take off AZT, 3 months, 4 months later, it can get activated, right? So latent TB, which is waiting for the right opportunity where we become weaker or whatever, can get activated. So these are the problems that are associated with TB. There are drugs, luckily, and these drugs are generally what we call as antibiotics. Those of you, anybody, I don't know, somebody in the family may have had it or otherwise, TB is a terribly difficult infection. You get fever and doesn't go away. You can take crocin, it just doesn't go away. And you're so weak from walking from there to here is going to be an effort if you have an active TB infection. So what is that? Basically, what kind of drugs you give? You give these drugs, which are nothing more than antibiotics, which inhibit various enzymes that are present in the TB. So what happens is when you start taking these drugs in combination, in about 2 months, you get to a point you feel amazingly good because you were in bad shape, perhaps. But the TB is not eliminated. It takes approximately 6 months. It is quite difficult to take a drug every day in the morning before you eat your breakfast and then wait for the 1 hour before you can eat. So somewhere along the way, compliance issues come. So what happens when you get compliance issues? Eventually, people stop taking. When people stop taking, the bacteria which was getting eliminated now starts festering and persisting, and it becomes resistant. Because why does it become resistant? Same concept. Everything can spontaneously accumulate mutations. Some mutations are going to make it resistant to the drugs that you are taking. So if you end up getting resistant to one of the drugs, it's called mono-drug resistant tuberculosis. If you get it for 2, it's called multi-drug resistant tuberculosis. If you get it for more than 2, which is second line drugs, it's called XDR, extremely drug resistant tuberculosis. And if you end up getting for everything, it's called totally drug resistant tuberculosis. Well, hopefully it doesn't reach there. So what is your solution? You have to continuously evolve and identify new drugs. Bedaquiline and delamide are the ones which are given as a final frontier. But how do we avoid this? Compliance is an extremely important thing. How many of you take the antibiotics that are given to you for 5 days when you get a strep throat? 3 days is mostly what people take, right? They leave it there after that. Am I right? 3 days. And what happens? It goes away, I agree. In some cases, in some individual case, it comes back. When it comes back, when they start off, they give you amoxicillin, okay? And you took it for 3 days, you stopped it, right? When it comes back, amoxicillin doesn't work anymore. So then they give you something called, I forget the name, but they give you a combination drugs. Okay, and these combination drugs is what is required for elimination. Means we are creating a problem for ourselves. Now at this point of time, forget the tuberculosis, antimicrobial resistance is a huge problem in all over the world, AMR. Why? Because now pathogens have evolved. I mean, pathogen is a regular bacteria, hospital-borne infections. People go for something called an elective surgery, they end up coming back with AMR infection. Possible, right? Because why is it happening? Majority of them are evolved over a period of time because antibiotics have been misused. People give antibiotics to cattle, give antibiotics to chickens, right? These things are used as food products, right? So essentially, you are getting antibiotics when you're eating also, and over a period of time, this is another way antibiotic resistance is emerging. So the problem is antibiotic resistance is a big deal if you are going to fight bacterial infection. Bacterial infection by itself is not a big deal, but you have to find, if you're going to not do compliance properly, it will become an issue. For example, in case of TB, drug susceptible TB is 83%, which essentially means 17% is not, right? And out of the 17, you make the 17 to 100, 54% are MDR TB, 30% are XDR TB. Okay, the highest burden of TB is in India, unfortunately, MDR TB burden. So this many cases, so eventually, if you have such kind of high drug resistant tuberculosis, you're going to have problems. What happens? The treatment period changes from something like 6 months to sometimes 1 year, 1.5 years, and that's pretty toxic treatment. And it's not easy on a patient who's going through this. How do you, not MTB, how does a bacteria acquire drug resistance? There are 4 common mechanisms for acquiring drug resistance. So what can you do? I take a drug. If I can modify the drug, then also a drug resistant. Or I can modify the target, I'll acquire drug resistance. The drug doesn't bind anymore. Because majority of the drugs are for enzymes. Enzymes do certain functions. If they accumulate certain mutations that would not allow the drug to bind, it's a competitive inhibition in most cases, then your enzyme is active again. So that is second. The third is intracellular concentration of the drug we can change, either by pushing it out or containing the putting it in. So all these 4 different mechanisms are used by the bacteria, and more mechanisms have to be figured out. Now, how do you do this? Again, technology comes into play. So what one does and what has happened is huge lot of information started coming out on what do you do. Basically, you go to the population, you take out those DNAs from the bacterial DNA and all, and eventually what you do is you basically sequence the bacteria and then go back and compare it with the reference strain, which is sensitive, and figure out what are the mutations that are accumulated in this bacteria, which is resistant. So information, for example, some of the mechanisms that are known, here is a drug called isoniazid. It gets converted into an active metabolite by CATG, an enzyme. If I get a mutation there, resistant. I get a mutation in the target, I'm resistant. Same thing here, target mutation makes it resistant. And that's true with many of them. Pumping out in and out makes it resistant. And changing the antibiotic in itself makes it resistant. So the multiple ways we actually can escape it become resistant to the antibiotics. In many cases, the antibiotic resistance is not determined. We have actually done some work, I would basically done by this girl, Sabha, but I'm going to give a gist of it. Basically, sequences are now available for the multi-drug resistant tuberculosis across the world. That's true for AMR also. So what you do is, again, this is beyond me. So other people come into play, and there are lots of programs, a lot of comparisons happen, predictions happen. So such predictions came with some new mutations that were only found in drug resistant strains, but not sensitive strains. So what we found is that some of these things, I really don't want to go there. Some of these things are in DNA repair genes. We ended up getting all the expected outcomes, like all known, existing set of mutations that are known to cause resistance were part of our shortlist. So we had confidence in our methodology, and we were looking for the novel ones. Hello, can you hear? Okay. Something has happened. You can be sounding. No? Okay. Can you hear now? Something I touched. It's gone away, no? Okay. It's giving it back here. So what I'm essentially saying is that we ended up identifying some novel mutations, just again, usual suspects, everything, in DNA repair genes. What is DNA repair? Every time our body replicates our DNA, we make mistakes, correct? Same way bacteria replicates, it makes mistakes. So DNA repair is to correct those mistakes. In addition to the mistakes, there is also a possibility like carcinogens and other things, which actually modifies the base, which actually while replicating changes the base pairing. So DNA repair enzymes recognize these mismatches and correct them. Without them, you will accumulate a lot more mutations in your body than otherwise. What we found is that Mycobacterium tuberculosis DNA repair genes or DNA repair enzymes were getting mutated in multi-drug resistant bacteria. So our hypothesis was that, I'll just not worry about it. Our hypothesis was that if you have mutations in DNA repair enzymes, you would end up getting higher mutation rates. Some of them are advantageous for the bacteria in whatever the stress we put. If it is antibiotic, it will evolve faster. And we proved this in multiple ways, doing biology experiments in animals and other things. But I really don't want to go into the research part of it. And because again, I'm trying to keep it general so that the audience are still interested when I'm done. So the second aspect is that now I want to bring out where the big data comes into play. So if I have to now talk about TB therapy, we have to find new targets, unfortunately. We have to eliminate the latent bacteria, and we also have to come up with alternate ways called host-directed therapy. But these are the aspects we continue to work on. So now big data and biology. All our genome can be sequenced in about flat 3 days, mostly. If you are willing to give around, I think it's around 1 lakh odd, we can get our genome sequenced. That easy right now. Whole genome of a person can be sequenced about 1 lakh. If it is a company, they might charge you a little more, but the bottom end-to-end cost is approximately 1 lakh. That is all it takes to sequence a human genome. So where is the advantage? Let us say you have multiple aspects, right? You have newborn screening can be done to figure out what kind of mutations they may have. And we already have enough knowledge on how the mutations contribute to certain phenotypes that we see. Sickle cell anemia. It's a mutation of one base somewhere, and that changes the amino acid and eventually makes it sickle patient. If you know that they have sickle cell anemia, there is a treatment for it. There are ways to deal with it. But if you don't know, diagnosis takes longer. So now next generation sequencing has actually changed the way we can actually do medicine, genomics, what we call as. So you can look into prognosis of therapeutic decisions, monitoring the disease burden, screening for diagnosis, and disease susceptibility can be figured out. Newborn screenings can be done. Even it can be done at a prenatal, may not be that wise in India because you get more information than what you're looking for. But nevertheless, this has all-encompassing applications. And what you're going to get is essentially basis, basis, and basis. Somebody has to make sense of it, and more of these bases are coming. There is another interesting project which I have not put here. For example, you go to archaeological sites, right? You find bones from 5,000 years old, 6,000 years old. We can extract DNA from that and we can sequence it. We get to know what kind of bacterial infections they have and what is their genomic DNA. Why does it impart? You would know where they are coming from, how the evolution has happened. So what are the origins and what is contributing to, who's contributing to what? Everything you can figure out just by based on the DNA sequencing or in sometimes even mitochondrial genome sequencing. So in total, you end up getting huge lots of information. Of course, it's very expensive to do this. I mean, we're talking about 1 lakh per one full genome sequence, but you have to put it in the context. In 1990, Human Genome Project was first time launched, and it took 8 years for them to sequence the first human genome. And at this point of time, we are talking about 3 days, 2 days, 1 day if you really have the machine with you, right? So information comes, but sometimes information cannot be made sense of, right? So this is going to be a process where interdisciplinary work is going to become important. Interdisciplinary with people having understanding. It's very difficult to have a complete understanding, but overall understanding is possible. Like what I'm trying to do is I'm trying to make my science easier for you people to understand, correct? Something like that can be done so that ultimately we come to a point where we understand each other's work sufficient enough to make a difference. So big data, and now that is with respect to genomics alone. Genomics only the starting point. There is something called functional genomics. In our body, in addition to genes are anywhere there, the methylation patterns, what we call as epigenome, methylation patterns change the expression of RNA. So there is something called epigenomics. Genomics is finding out the sequence. Epigenomics is finding out the modifications in that. And then you get transcript, which is RNA. RNA can be spliced because we get full RNA, which gets chopped in, ligated to become a mature RNA. RNA gives you, if you go with just RNA alone, the genes alone, we have 30,000. But in now, over a period of time, people have recognized that RNA can be alternately spliced. Some exons can be taken out. Means you have pieces with in between sequences are taken out. They are called introns. The pieces that come together are called exons. If I skip an exon, my sequence is different. That's called alternate splicing. That variation is there. And RNA gets modified. And that changes the stability, that changes the expression pattern, that changes many things. So all these things are big data. And all these things are we are putting into the machine, somebody is making sense of it. And they all have an implications on the phenotype. What makes you, you, right? And then you have the downstream end, proteomics, the changes in your protein levels. The changes in the protein levels can give you a disease at times. The mutations in the protein, of course, will give you certain major flaws. And then there is something called metabolomics. Metabolomics are the metabolic changes that happen in your body, which is also dependent on our lifestyle. So all these things can be, total thing is called functional genomics. And all these things are so much data that it takes huge level of combined effort of many people to make sense of things. This is where we are going. That is a reality. And so I'm not added one complexity here. There is a title that I read somewhere. Are we more bacteria than humans? Because we have 1.3-fold more bacterial cells compared to our own cells. Right? 1.3-fold, whatever, 10 to the power. So bacteria is more in our body than us. So we are 1-fold, they're 1.3-fold. That's where we are. And the interesting part is the microbiota contributes to many things. For example, a child born through regular birth versus C-section. There is a difference in the microbiota exposure. That's known. And that contributes to certain things. So this is an interesting aspect. Microbiota has become a big deal in the context of now human health. Microbiota means you can figure out the microbiota by doing something called metagenomics. Microbiota contributes at times, microbial population in your gut contributes sometimes certain metabolites, which are important for your digestion to a lot of other factors. So this is another complexity, and more big data will be generated. Earlier, people were looking at things independently, microbiota separately, genome sequence separately. Now, general concept is you need to have a holistic view. Everybody talks to the other. Means host and microbiome and the pathogen, they're all in one sense talk to each other. The severities and what happens and the consequences, we are still not there to predict it. It's not like I can sequence a genome and I can tell that you're going to have severe infection if SARS-CoV-2 sees you. We're not there yet. But I think we are moving in a direction where such kind of predictions are going to become norm. Life is not as easy as one would like to put it. It's not black or white. Mostly it's in gray. So we may be able to, like, for example, breast cancer, right? In 33% of the cases, women who have a BRCA mutation would develop, or 33% of the women who have breast cancer have mutations in BRCA. But not the remaining 66%. It's all percentages, right? But these things can be predicted to an extent. Now you have a mutation, you may be prone to it, right? So that can be predicted, but would not have been possible to predict, say, 20 years previously. So this is where a lot of integrative work between various disciplines is required for us to achieve these goals. And I personally believe Hyderabad provides that opportunity for all of you and for us in a way to come together and work on relevant problems. There are problems that are relevant to human health and make a difference. Thank you.
Thank you so much, sir. So we'll open the floor for audience questions. We have some mics going around. So we can start. If I can start with the first question. So we are given to understand that viruses. So viruses don't really have an agenda for killing us. They want to replicate as much as possible.
Survive.
Yeah, they want to survive. So the fundamental question is, why do we end up dying? Isn't it better for the virus to just replicate more and spread it through me, for instance?
It's not their fault, it's their host is weak. So, as I said, when you start talking about the original SARS-CoV-2 infection, a lot of people died, I agree, but a lot of people survived. If you look into the survival rate, it's pretty high.
Right.
So over a period of time, the evolution has gone towards Omicron, in one sense, a milder virus. Correct?
Right.
So it is, host has to survive for them to survive. So there is no point in killing the host. But it so happens that the host being weak is not fault of the virus at times.
Right.
Correct?
Yeah.
It's survival of the fittest. If somebody is injured in a herd of whichever, whether it is a deer or anything, they don't survive. Correct? Unfortunately, this is the truth of, so in one sense, when virus comes to us, it wants to survive, it is evolving, it is only looking at its own survival point of view. But unfortunately, the point is we cannot talk about the variations of the host. We are varied, we are different, we are not the same. Some of us have diabetes, some of us have hypertension, and some of us have both, and some of us a lot more. Right? And in addition to this, and so these things contribute to how you actually, some people, despite all these things, have really had no major symptoms. So this part is still something that we would like to understand. I'm sure the world likes to understand why some people showed major symptoms, some people not so, and how much comorbidities have to do with it and stuff like that. So yeah, that's how it is.
Yes. Questions from the audience? Sure.
So yeah, my question is again on COVID only, because like being a person who has been infected with COVID once, so I still face a few of the side effects that I did have at that time. And when you were talking about HIV, you did say it would go into your genome and mutate a few things in the.
Not mutate, it survives.
It attaches itself to that. If COVID does something like that as well, and is that why I'm still facing side effects? Does it?
No, I don't think you would even have any COVID if you today do a test on your nasal this thing or whatever. You would have any SARS-CoV-2 in your.
Yeah, if I test negative,
you will be most probably negative. This is currently called long COVID, and it is still not properly understood. Understand from the context of science, it's a very young disease. Okay. It's 2 years only, and more data will come, and people are working on these aspects to understand why some people have long COVID and some people have relatively no asymptomatic and nothing much happens. These are the aspects one needs to understand. Okay. There is no, most probably it is the way your cells and your body kind of got damaged during when the infection has happened. But long COVID is still not properly understood at a scientific level. So to give you a definitive answer, it will be difficult for us at this point of time. Over a period of years, we will have definitive answer. Not necessarily from our work, but from the work that happens across the world.
But is it a possibility that it could have mutated, it could have attached something to the genome?
There was one report I recall which they talked about COVID kind of can get integrated into your genome, but really not much I've seen on that line. Okay. So I believe that it is maybe to do with our body, the way it has faced and it survived or whatever. and now some of the things that are that thing. We need to understand this better. So that's what I'm saying. No, never give a definitive answer in science when you don't have all the information.
Sure.
In any case, as I said, central dogma was considered it's written in stone in 1950s, not anymore, right? So it keeps on changing. It's an answer will be different with time.
Yes.
Yeah,
But long COVID is actually there. What you call as COVID side effect.
yeah, my question is around vaccination. So generally, how does vaccination work? And today we are hearing like the vaccination, what we're taking, like it's 6 months duration it work, and then we have to go for a booster, and then booster works for another 3 months. So
It's
what
like-
exactly it is?
It's like this. When you vaccinate, essentially you are telling your immune cells, here is the enemy, recognize it, right? Then you have two kinds of response. One is obviously the antibody response coming from the B cells, which would basically be circulating in your body, which goes over a period of time. And then you have the memory, certain level of memory to recognize the enemy next time they come in. What happens is if you get infected, you may get a fever, but eventually your memory kicks in. It produces both the T cells and the B cells and the antibodies required to kill it. But not every vaccination gives you the similar levels of memory response, right?
Okay.
And so in case of COVID, we realistically do not know how long the memory, generally antibody levels start going down in about 4 months. By 6 months, your antibody levels are not anymore there. And now the question is how long the existing vaccination memory lasts. As of now, they're thinking maybe every year would be a good idea, but it's all right now, I would say speculative. You need to do systematic study to figure out how long the vaccine memory stays. In the sense like, you have to do a study after 1 year, what kind of infections people are getting, what is the gravity of the infections. Is it going to be like flu? In case of flu, every year you have to take a flu shot. Every year. So, and if the variants come, so you have to take for sure. The previous year's flu shot doesn't work.
So it's in progress, the research in progress.
It's something evolving, but I think since people try to be on the cautious side, they'll say it's not a bad idea to take every year once, right? But is it really required every year? What kind of vaccines are required? What kind of, like over a period of time, even other aspects will come into play. But every year vaccination is one thing to say, okay, I'm going to boosting it all over again.
So what if we proactively take before our duration ends?
So
Is it harmful?
So for example, let's say you have a vaccine which was with the previous variant and you're taking that as a booster. The antibodies that are generated with the previous variant may not realistically neutralize you against the new variant that's coming in. It gives you other kind of responses which will certainly take away the severity, but it doesn't completely protect you from an infection if the new variant escapes the antibodies that are raised against the previous variant. So infection per se is not bad there, and it's not even the end of the day. It's the end of the world, but these things are going to happen. So vaccination protects you for sure from severe infection and hospitalization and certainly death.
Thank you. Sir, thanks.
Here. Yeah. So I have two questions. First one is like after a lot of science and technology research, we are coming with the vaccine, but this virus is slowly overpowering us. Okay, again it is coming. So like not a living organism, but taking shelter in us.
It's mutating in us only.
Yeah. So now is this journey for coexistence or survival of the fittest?
I think it's coexistence. Eventually, we are going more and more towards coexistence. And the general, if you start talking to virologists, which I am not, and you look into the news, they talk about eventually reaching something called an endemic from pandemic, where certain areas the spikes will happen, but not the whole world will be facing it at the same time. If you now go, for example, and look into the world COVID map, it does not come down. It's still going on in various parts of the world. But in India, it has come down. So we are going to have different peaks for different countries. Hong Kong is really taking the hit currently, and it'll come down after a while, right? And this continues to happen. And we have progressed. That's why the death rate has come down. You have to appreciate that aspect. People were heavily dying. When the originally COVID came, so many people died because it was a fresh virus. Because we have vaccination and we also have prior exposure now. We are in a better situation than where we were.
Hello, this is Krishna Reddy faculty here. So we are reading a lot of articles about climate change, environmental problem, deforestation. So is it science actually? Why they are writing such articles in the newspaper saying that this is the reason for COVID? Is there any correlation of deforestation, climate change on COVID, or it is a nonsense?
I have no clue about
Because
the
what you
climate
have
change
talked
and
about
COVID.
is it is a natural phenomena, you know.
I would
So-
say natural phenomena. I'll tell you, in general, viruses. Also, there are viruses that are present in various animal species already. I mean, they are their host originally, and the species jumps happen. What we call in our this thing called zoonotic transmission. When we give it to lions, it's also a transmission of a reverse kind. Correct. So viruses jump, and it happens, and now, if you want to correlate it with the climate, I'm not the right person to talk about it. And yes, climate change by itself is a serious issue. And we know the implications of the climate change on seawater levels, temperature changes, which obviously would have implications on how microorganisms also survive. In addition to us, like in a higher temperature, sometimes may make it better for the microorganism to replicate and stuff like that. But I realistically am not in a position to make a scientific comment on that.
Yes. So I have a question about like the exact nature of viruses, or like the fundamental nature of viruses in general. In school, we were taught that viruses aren't exactly considered living or dead, because the living aspect is they have the tendency to replicate themselves by infiltrating cells, but at the same time, they show no other tendencies of no other characteristics of conventional living creatures. So, based on your experience, what would you consider it as, and is it a valid statement that viruses are
Virus,
not?
see, the reason for that is simply because it cannot survive on its own. Like you take a virus and put it in any kind of a media, like say any kind of a media, whatever. So for example, I take a bacterial cell, tuberculosis. There is a contained certain media, autoclaved proper media. I put the bacterial cell, it replicates. The bacterial cell is self-sufficient to make double of itself. Virus cannot make that or cannot do that in any kind of a solution. It can only do it when it has a host. And the host changes. For example, bacteria have viruses called bacteriophages. And that is so specific. What is going to go to Mycobacterium doesn't go to E. coli. So virus only replicates and thrives or replicates or survives in the host. By itself outside, you cannot culture it. So that is why the concept of it's not exactly living, and it is certainly not dead. It needs a host. That's the concept, nothing more. So what do I consider? Viruses by itself cannot replicate. So are they living? Not really, if you consider that way. Independently cannot survive. Both thrive inside our bodies, are not exactly our bodies. It can be bacteria, which is its host. It can be anything which is a host. So there are viruses for everything. But they thrive eventually, or at least survive.
All right, we have time for one more question. We don't want to bug him with too many questions. So,
Hi, sir. I'm
Yeah,
Ayush,
thank you
a
very much for
PhD
the
scholar
presentation.
here.
So I was,
okay, let's have two questions. You can go, and then sir, you can later.
I just wanted to ask, is there any role of this new gene editing technologies, this CRISPR-Cas? Maybe we can remove some of the mutations in the
virus.
Not in the, maybe in the bacteria. It's, I don't know whether it is.
Okay, gene editing technologies. There are two aspects of gene editing. One is that either removing or adding a base. But the second thing, what you were talking was gene therapy, actually, where you actually modify the genes of the host.
New this CRISPR-Cas technology.
Yeah, CRISPR-Cas. Yeah, CRISPR-Cas is basically, I'll tell you to just elaborate. There are proteins called Cas9, which originally were part of the bacterial immune system, essentially. They recognize if you put a certain RNA, guide RNA, or some way, they actually can use the technology to modify our genome. That's what he's basically talking about. Modifying our genome is editing. You can edit our genome to add bases, to remove bases, to change the gene itself. And this one has a huge potential. Specifically, let's say somebody has rare genetic disorder where a particular gene is not functional anymore. If we can actually evolve this process to such a level that we can change the gene and put a functional gene there, it will certainly give you a huge health value. May not be from the context of the viral infections, but it is per se has huge potential. Are we there yet? Not yet. It's a work in progress by many labs.
Yeah, but if we can modify genomes of tuberculosis, let's say, that's what I see.
We modify the genomes of tuberculosis a lot. That's not difficult to modify, including CRISPR-Cas9. But in CRISPR-Cas9 doesn't work effectively in tuberculosis. We have something called gene replacement methods, and we can do a lot in terms of tuberculosis manipulation because it grows outside. But even if you end up getting a strain which is not so virulent, then what you're talking about is using it as a vaccine. Even those efforts are going on. So those kind of efforts are on. But with respect to modifying human cells, they're a little more complicated because you have to worry about the other aspects, safety aspects more than anything else. And I think there is a lot of work that is going on in this area. There's a lot of work that is going on. Again, I'm not an expert on it, so I realistically don't want to venture beyond a point, right? Or at least I don't have the literature knowledge at this point of time offhand to give you a proper answer to how far these kind of technologies are progressed. But it's something that people are working on.
Thank you.
Hi, sir. This is Ayush. I'm a PhD scholar here. Thank you for the wonderful talk. So I wanted to ask something connected to what we discussed about antibiotic resistance. We have heard a lot about misuse and overuse of antibiotics and antivirals during the treatment of COVID in India. It was also attributed as one of the cause for rise of black fungus cases at one point of time after second wave. So I want to ask you a more generic question, that how do we ensure that the kind of knowledge probably that we have today at CCMB or any other research oriented labs, that knowledge reaching to the physician who's at the end going to treat me as a first line of defense? Because what best I as a patient can do is to trust my physician in terms of whatever he or she is prescribing me. So as the director was also talking about inculcating that research culture and right at the level of bachelor studies, do we need some kind of academic policy intervention in medical studies also, such that we can tackle these kind of problems?
Couldn't agree more. Really, I believe, and we have a program at CCMB for medical students. They actually can come and work in the lab for 2 weeks or so. So this is one sense gives them a view of the other end of the world, but it has to become integral to many of the medical things also. It is good to know about many things, but it's also important to have a better knowledge of how these things work, the research end of it, and get a better idea at a molecular level. It is something up to the policymakers. I mean, really speaking, but is there any advantage? Yeah, huge advantage. It's like I'm telling you guys, who are in engineering line, that there are all of us need to work together to solve. Same way, eventually that is true as much for the other end, the medical. In US and all, for example, MD PhDs is a norm. A lot of MDs get into PhD and eventually they are researchers also. Not as much in India, but I think we would ultimately go there. We are going slower, but I'm sure we will go there. And to some extent, it's a policy issue.
All right. With that, we should once again thank Dr. Nandipuri. And as a small token of appreciation from IIIT Hyderabad, we'd like to present this. Thank you. All right. after that wonderful discussion, I think we should again direct our focus towards the R&D showcase that we have assembled. To introduce it, I would like to bring on stage Professor Professor C.V. Jawar, who is the Dean R&D of IIIT Hyderabad.
Thank you, Aftab. Aftab rightly put we have to suddenly remember we are in the R&D showcase. We are in a completely different world till now. Learning about something really contemporary, important, basic science, basic a taught in a way that we all understood. Thank you. So R&D showcase has been a regular event of IIIT, as Professor P. mentioned earlier. We were waiting for it when the COVID happened, or when this wave came. Everything was new. So I just want to go back a little bit more. I think possibly the first time we did an R&D showcase was in 2002, 20 years back. And we almost did it every year, except the last year, the 2020, possibly where we postponed. And 2021, we went and did an online way. And today we are almost back on with the physical event, at least in a very limited form. But a lot of things have changed from the 2002. So if I try to remember what was going on in 2002 to 2022, 20 years. I still remember the days in 2002 where students will be struggling to set up their computers in different classrooms with possibly no power cable there, no network connection with the LAN cable getting wired, and code getting debugged in the machine what they got to demonstrate it because some mismatch in operating system, mismatch something, a lot of issues, practical issues. And we are all in the night, there is a worries, will this demo work tomorrow morning? So over time, I think today things have changed a lot. So last 2 years have seen 25, 30 years back, students come with a laptop. Demo is set up on my server. I just need a browser. I need only a Wi-Fi. Or I have a data on my phone that works. So systematically, the way we were demonstrating our technology changed. You can see a change in the way we presented our result. Research itself, the definition of research changed a lot over time. At IIIT Hyderabad, there is no need to tell that research is not about just writing papers. The demos, starting from the first courses here, people are encouraged, if not forced, to demonstrate their results, projects on real data, doing live demos. This has gone to the roots, so that the students are not afraid of writing code that works. It is no longer a code and show that yes, I have a result. So releasing code to the public, that became a culture. Just like publishing papers, people release the code or contributing to the open source projects. There is a lot of encouragement on having the result which is useful for others, the result that others can see, others can experience, working on problems that matter to the society at large, working on socially relevant problems, working on problems that are interdisciplinary. So people are not possibly averse to work on problems touching discipline that you have never heard. So I was when I listening this talk on biology, I was just thinking about maybe a few years back, if such a talk was given, I would not have seen people attending with this level of passion on this campus. People with trained in maths and engineering possibly were reluctant to listen about biology. I think this is the right time, possibly. We need to get biology or the disease biology or computational biology or bioinformatics or whatever we talk, possibly into the roots. This has been always the way IIIT, if you look at looked at interdisciplinary research. Our students who are trained on, say, programming. So today there is no need of telling about how to program or what to do when we do research. It is taken for granted because of the coursework, the curriculum, the way it is taught. In the next level, people talk about system building, building demos, building system, building end-to-end things. There is no need to teach people. Students are well-equipped, powerful arm. And possibly similarly for a lot of AI, machine learning type of tools today, half the population of IIIT may be taking such courses. And they're equipped with the terminologies of contemporary tools, techniques that can be applied to various areas. Maybe very soon we should look at things like IoT or computational biology or disease biology to be reaching there, so that just like half the population is vaccinated, we see the difference in the responses. The moment half the students on campus take such courses, it gets into the roots. The research happens naturally, interdisciplinary research. I think these are things that we can keep in mind as we move forward in these sort of areas. Research, not just talking about the research that gets into demos. The next thing that happens possibly is about now, how do you take this to outside world, work with startups, work with end users. So we have very vibrant, a startup system here around, encouragement to work on that. So I look for a day where when we have this R&D showcase inauguration, half the population in this room are the alumni who are doing startup here or around, or working on problems, taking technology forward beyond just looking for a regular job. I see the trend, I see the systematic change happening, and maybe that is what we are going to see in the next few years. So today we have an open house or R&D showcase where a lot of things that happen in the lab is kept open. There are a lot more possibly which are not being displayed. Feel free to see, interact, talk to people, talk to students, talk to faculty members, online, offline, campus, wherever, whatever form that you are comfortable with interacting, and continue to, maybe there are contact addresses, continue to engage, continue to contribute, and strengthen and let this be a mutually beneficial. So we have around 300 posters also, or demos and posters on display from around 20 research labs. And this is available here today and online remains for more time. So we first time when we tried online last year, we had seen that it was kept open, and we have continuous visitors from all over the world for the last 1 year, which possibly we did not even know. We did not even bother or monitor that level. So there's so we are keeping things open, and this openness should remain forever. We want more people to come, use our results, and take it to the next level. So I encourage all of you to visit all the posters, demos, interact, and contribute, work together and use this as a forum for enabling that. Thank you. I'll give back to.
Thank you, sir. So in the spirit of encouraging new age technologies, we have what we call a center spotlight, in which we typically put the spotlight on two of our most exciting new research centers, that are tackling some of these problems that we have discussed. So to talk about one of the centers that we have is the Center for Quantum Science and Technology, CQST. To talk about that, I would like to call Dr. Indranil Chaudhary.
Thank you, Aftaf, for this kind introduction. And respected chief guest, Dr. Binay Nandikuri, our respected director, Professor P.J. Narayan, our Dean of Research, Professor Sivjit Jahar, and our Dean of Academics, Professor Kishore Kathapalli, and our registrar, Professor Pradeep Kumar. And all the my colleagues, all the participants, and all the students who have joined us today. A very good morning to all of you. I'd like to take this privilege on behalf of my colleagues to tell about this new center, newly formed center, as Professor Atta was mentioning, Center for Quantum Science and Technology. This is something that, this is a kind of a dream that we have cherished for many years, and we came into reality in the last year and we are now a full-fledged center. And all these things happened because of the constant encouragement and support from the institute, who had actually guided us in a sense to create this center. And to tell about the center, I would first like to tell about a little bit of the timeline of the center and the people who are there in the center and the area of the researches that we are doing and what we'll try to look into future as a center, as a collective unit. So regarding the timeline, so this is something that this area of quantum information and computation was taught by my colleagues like Professor Srinathan and Professor Harjinder Singh before I joined here, as an introduction to the students and in their regular courses. And this had actually ignited a spirit among the students to pursue research in this area. Later, what we found that many students actually took this as a subject of research and did their master thesis. Now, all of these students, after graduating from here, were placed in various premier institutions across the world, and they did their PhD as well. Now they are doing their postdocs, and some of them are doing their postdocs right now. So, there has been a culture of introducing the topics to the students as a research topic. Not only that, there are other also courses like based on this area that had been taken by us. But now for the last 2 or 3 years, we slowly started building up this group who are by taking actively faculty who are actively working in this particular area. So right now we have a very strong and a dynamic group who are doing research in this area. This includes young faculty like Dr. Shamadev Bhattacharya, Dr. Shubhadeep Mittal, Dr. Shantanam Chakravarti, and Dr. Siddhartha Das, and experienced faculties like Professor Harjinder Singh, Professor Kannan Srinathan. So as a combination, we had combination of both young and experienced people in the group. So as a research areas, we also try to work on different areas like quantum computing, how quantum computing, quantum algorithms, quantum computing, how this quantum computing is going to change the way we are doing research, doing computing and information processing currently at this moment. So and there are other areas like quantum networks, quantum information processing, quantum cryptography, how the idea of the cryptography, classical cryptography, will be changed with the help of quantum cryptography. So these are different areas which we are working on. And also as a group, we try to build up a very supportive and ambient atmosphere within our center, in the sense that we will be able to create more students, more manpower and human power in terms of the PhD students, in terms of the master students, and also in terms of the number of publications and the projects and the research that we are doing. So as Professor Jahar was mentioning, the research has changed for a lot in the true sense that it is not only associated with publications, but also building up an ecosystem with the industry and creating a synergy with the industry and the other, taking the impactful problems, and so that we can actually contribute to the society in strong sense. So with these aspirations as a young group, and we want to become one of the best. This is right now, it is the most largest group in the country in terms of the number of faculty that we are having. So with this aspiration, we want to bring one of the most best centers and the best research centers in the country, if not the one of the best centers in the world itself with the years to come. So with that, I thank once again Aftab for letting me giving the introduction to the our Center for Quantum Science and Technology. Thank you. Thank you.
Thank you, Indranil. That definitely sounds very exciting. So the next center we have on our center spotlight is called the Computer Systems Group, CSG. And for the introduction for that, I would like to call on stage Dr. Suresh Purini.
Thank you, Aftab. Good morning, Dr. Vinay sir, Professor PJ, and Professor Jawhar, and good morning, everyone. So I am one of the members of 4 members of Computer Systems Group. Myself, Dr. Deepak is here, Dr. Vignesh, Dr. Tejas. All of us put together, we work on different aspects of systems, right from actually building stuff to performance modeling of how to model a system and try to come up with optimal performance points when we build these large scale systems. So if you think of modern computing systems, these modern computing systems are very complex. Like if you take your laptop, it has multiple cores, it has a very complex memory hierarchy, and it has multiple accelerators, GPUs, and if you go to server scale systems, you will have FPGAs and lots of things are there. All these things, they give us huge amount of raw computing power at our disposal. Okay, and if you move from one machine to a cluster of machines, then this raw computing power gets multiplied by 100x, 1000x, etc. How do we use this raw computing power? How do you translate this raw computing power into application performance gains? That is the key problem. That is one of the key problems that we work in our center, translating huge amount of raw computing power into application performance gains. So with respect to this question, I'm very happy to thank our guest speaker for setting up the stage, because bioinformatics is one of the places where there is huge amount of many problems which require lots of computation, lot of data, and lots of algorithms are there. And these algorithms, they are not linear. They don't have linear time complexity. They are no super linear or exponential time complexity is there for most of the problems that we encounter in the bioinformatics space. So one of the big areas where we work in is in how to solve these problems from this bioinformatics space using this available computing power for us. So that is one area of research in CSG Center. The second area is, of course, AI/ML. So like, for example, think of a situation, let's consider Hyderabad. In Hyderabad, there could be at least some 50,000 CCTV cameras there distributed across the whole Hyderabad. Minimum of 50,000 cameras, CCTV cameras will be there. That means 50,000 CCTV camera streams are being pumped to the data center. How do we process this voluminous amount of data? A huge amount of data being streamed. How do we process it? It's a very, very challenging problem. How do you store it? How do you analyze it? How do you elicit interesting information? Do query processing? All these challenges. It involves, like a multi-pronged approach. It's not like, for example, interesting questions such as, should I process this video stream partially at the camera itself and then send some information, interesting information back to the data center? Or should I send it to some intermediate fog node? Or no, so what is the right architecture I should come up with? In this multi-tiered architecture, we have the edge devices, and we have these intermediary fog nodes, and then we have this data center. And there is this whole lots of video streams, lot of sensory data, all these things are coming in. How do we architect our system so that we will be able to process all this information and be able to provide useful, actionable information back to the end user. So this is the second area of research we do, large-scale distributed video analytics and data analytics. This is the second space. And the third space is we work quite hands-on at the hardware level also. Like, for example, like there's lots of AI ML all over the place. And even in this, this video processing, you have to do lots of inferencing tasks on these video streams. And so if you just do it on like regular CPU cores, etc., what happens is the amount of power consumed will be substantial. And so power is a very important thing when we think of edge device or even a fog device, or even at the data center level, because of the sheer amount of the compute density at the data center side. It's very important. Power becomes a very important factor, both at the data center side because of the compute density at the data center side, and at the edge side because the resources are very limited. So power, you don't want to drain out the battery. So in this context, so we have very interesting works where, like, for example, we can take a machine learning model. So let's say you have a YOLO model or any kind of machine learning model you have. We have a compiler which automatically synthesizes hardware given a machine learning algorithm to this compiler. And it's a very challenging task, like taking a machine learning model and automatically synthesizing and understanding the structure of computation in that machine learning model, and then synthesizing a hardware accelerator which performs really well based on the metrics such as performance per watt. There are quite a number of challenges that we have to solve during this process, right at the architecture level, right at the compiler level, and at other places. And then so we also work on, like Professor Deepak works on safety critical systems. How do we design, like systems which fault tolerant systems, robust systems for safety critical systems like automotives? Because computing is deeply embedded. Like when you hit your brake, some process might be invoked, and if the process is not scheduled in time, then you are in trouble, right? So so designing robust systems, verifiable systems, systems which give guarantees for in the safety critical infrastructure, a lot of work goes in there. And so overall, so while all these things are very hands-on, lots of algorithms, understanding of architecture, systems, network stack, all these things are there. It's not like we do these things in a blind way. We need to use the right performance modeling tools, right queuing theory tools, in order to understand and design these complex systems so that we hit the optimal performance points. And also in this space, when there are large number of small edge devices or large number of agents working together, there is both cooperation and competition that kicks in. And whenever there is this cooperation and competition kicks in, then we need to analyze that system properly through the use of right mathematical tools such as game theory, etc. And Professor Tejas, he is an expert in game theory and applications of game theory in analyzing systems. So one thing I missed out is in this novel space with large number of edge devices and fog nodes and data centers, the way we think about in network also, it could, we may want to rethink about networks also. And Professor Vignesh's expertise is there, where he has a novel way of thinking on by what are called as information-centric networks. So overall, like if you see in our center, we do many things from hardware to networks to process using processors and compilers, distributed systems, lots of stuff happening in our center. We are so much excited. All of us, we work together as a team, and we would like you to join us, or to hear more about what we are doing and be part of this exciting journey going forward. Thanks a lot. Thank you.
Thank you, Suresh, for that wonderful introduction. So that actually brings us to the end of our inaugural session. And because we are only 26 minutes ahead of time, I'll take 2 more minutes. So I want to play a video game with you guys. And people who know me well know that I'm an avid gamer. So let's see if I can share this. Yes. It's trying to. I started sharing. Yeah, there we go. So this is IIIT Hyderabad in the metaverse. Okay. And I can basically move using my cursors. You can see all the centers here. They, all the center posters are here. And the interesting thing about this is that if you enter that blue line that is there in front of the center, you can actually converse with people who are also there. Okay, so it's a distance aware metaverse. Okay, so people who are next to you, close to you, in that blue, inside that blue line, those are the people you can converse with. Okay, and we have these these posters, these stalls, so to say, in the metaverse for all the centers that we have here. Okay, so for that you need to download this app. It's called the Next Meet app. It's like Pigeon introduced as well in the morning. It's one of our startups in CIE, so we are also encouraging them as well. We are very excited about it. So this is our first metaversal R&D showcase. So with that, I think we can close the inaugural session. I would like you to I would like you to leave from these doors, exit from the front doors, and if you exit from here and take a right, that is where the KRB auditorium entrance is. That's where our registration desk is. So you can start your journey from there. You will get agendas there. You can get the map of the distribution of where the centers are physically located, and all of those things can be provided to you. Thank you so much for joining us this morning.